Hello everyone, welcome to another episode of High Alchemy. Today I have Dr. Jerry Lanchbury, the former chief scientist at Myriad Genetics, one of the largest clinical genetics company in the world, as well as the, I believe, first member of Nucleus' scientific advisory board. We are so excited for this episode, and I cannot wait to talk about everything politics, genetics, and of course, Nucleus. Jerry, how are you?
I'm great, Kian, thanks.
I think you flew out from London for this, or where were you? Not in London, you were in, where were you?
I was in London the previous week, and then I was back in Utah, and I flew out from Utah.
Okay, wow, so I hope it's going to be worth it.
It's definitely going to be worth it.
I know we're going to give the fans what they want. I know it. I mean, you have such a unique kind of, I think, not only experience and technical knowledge, but also just such a profound, I think, ideology when it comes to genetics. And I think one thing that's always been very interesting about you being on the scientific advisory board is continuing just to push Nucleus to new heights. So I want to say, first and foremost, thank you for that.
Well, you're most welcome. I'd like to thank you for having me on the podcast. And it's a real honor because you've had some extraordinary guests, and it's an honor to be here in their place.
Amazing, Jerry. My first kind of question for you, if you will, is sort of tell us a little bit about your childhood. Tell us about how you got into genetics. I think it's always good to ground these conversations in who are we talking to today.
Yeah, I'll give you a few vignettes because I could talk for this about this for ages. But I had a pretty unusual childhood as a consequence of my father being in the Royal Air Force in the UK. And he grew up on a really small island called Guernsey, which is and isn't part of Britain. It has a strange relationship. It was 12 miles by 6 miles. And at the age of 16, he really wanted to get out of there because options were limited and he didn't want to be a crab fisherman or a bus driver. Joined the Air Force, eventually became an officer in the Air Force, sort of worked his way absolutely up from the bottom. And he was an electrical engineer. He was an extraordinary electrical engineer. And I sort of discovered in my mid-childhood that his job was essentially to work on a team to vaporize command and control centers in the Soviet Union. Wow. He was nuclear bomber crew on the Vulcans, which were the high-altitude reaction force that worked with the United States to ensure the security of Europe and the US. And so it was a strange life because we moved all around the world from place to place. He was around some of the time, but not all of the time.
Could your dad just tell you what he did? Or was it confidential?
Well, you know, when you live on a bomber base at the end of the runway, and I knew he was on quick reaction alert, some of the time, which is when they have to get the bombers into the air in a matter of minutes. You know where he is. They're eating in the mess and they're staying away from home. And then the alerts go, and it's 4 o'clock in the morning. There's huge siren noise, and the bombers start blasting off. And so you know what their job is. It's perfectly obvious. And so I kind of lived that life until I was about 11. Went away to boarding school in Norfolk and had a very interesting sort of educational childhood. I had some ropey periods, some rough periods. I kind of left school on the morning of my final exams. I was homeless for a while.
What? Wait, really?
Yeah.
Okay.
I was homeless for a while. Did all sorts of things.
How'd you end up in genetics?
Well, eventually I went back just to take my final exams about a year later. I hadn't been to school, didn't know anything. Didn't do spectacularly well, and then scraped into a university, who I'm very grateful to for having me, and then really turned my life around. Graduated top of 2 of the courses, 80 people, got the biology prize. During that education, I got exposed to human genetics.
And what college did you go to?
I went to Aston University in Birmingham. So it's the second college in Birmingham, UK. During that, I got exposed to human genetics, and I think via a lecturer who was called Dr. Martin Cole, who was a fascinating character. He was actually a plant biologist, but he lectured in human genetics, and he also did one other thing. He ran the first surrogate sex clinic in the UK in Birmingham. And so this led to all sorts of amusement. And I'm sure this doesn't happen at Harvard or Yale. But, for example, there was one morning when he was on the front page of the Sun tabloid newspaper, and they'd put some cartoon crown on him. And the headline was, sex king coal in 4-inch banner headlines. And so we had Human Genetics. So, the 30 of us all went out and bought a copy of The Sun. And as soon as we walked in, we all took it out and sat there with Sex King Cole emblazoned across the entire classroom.
Oh my God.
And so he was a Falstaffian character, extraordinary man, and controversial in his way, in his time. But the thing that I started to learn about at that point was polymorphism, the incredible diversity of human populations, the human species. And, in a world where much of the political focus was about ignoring differences between people and, goodness gracious, certainly not admitting that any of those differences might be due to underlying genetics, I suddenly saw this great panoply of variation and difference in human beings that was part of their inheritance, part of what they had taken from their parents. I was absolutely fascinated and blown away by it.
Okay, so what year is this approximately? And it seems like you take this class, there's this professor, you become fascinated by human genetics. What then? What happened?
Yeah, we're talking about 1980, when my realization of the degree of polymorphisms in humans.
And is this like junior? Is this like, what is it, sophomore, junior?
Well, I went in 78 and finished in 81 because we do 3-year courses.
Okay, got it.
So it doesn't quite align with the US system.
Got it. And then you went and got a PhD?
Yeah.
In human genetics? Or?
I went and got a PhD in human genetics. So I have a PhD in human population genetics. I got there in a sort of roundabout way, but I ended up with a Science Research Council scholarship from the UK government to study population genetics. And when my supervisor was Surinder Singh Papier, and he was a Sikh, and he had really strong connections to India. And when I went, he said to me, Jerry, there's 2 things we can do. He said, you can work on statistical population modeling, and I really don't know if that's going to lead to a PhD, but you can take the risk. He said, or we can go out in the field, and so going out in the field meant spending months and months and months in India, going to leprosy clinics, going to tuberculosis clinics.
You chose that one.
I chose that one.
That sounds like you. You're adventurous.
It is me.
Yeah, you like to travel.
I do. So we encountered tribal groups in northern and
what was like the research question? What was the fundamental investigation?
There were 2 questions really. One was, what's the degree of diversity at the genetic level in India? Are there degrees of inbreeding? Can you detect selection? When you look at tuberculosis patients with the genetic markers that were available at that time, can you pick up any genetic markers that are population level or associated with disease?
Were you running microarray in these patients?
No. Okay, so let's go.
What year?
Let's go back in history. Imagine you started a PhD with access to 40 genetic markers across the genome, which is where we were in 1981, 82, 83.
Yeah, and just for people who have context here, right? Human Genome Project's in 2000, right? So to a very real extent, genetics was in its absolute infancy.
It absolutely was. In its infancy, but at the point where a lot of people had worked very hard for a long time with blood groups, with red cell enzymes, with serum proteins. It's just we didn't have any DNA markers. That era had not arrived yet, and really the era of DNA markers arrived about the time that I was finishing my PhD, and that was the time of RFLPs and gels and restriction enzyme, digestion of DNA, sticking that on a gel, electrophoresing it, and then coming in with radioactive probes, usually cDNA probes, partial cDNA probes. Then it flipped to oligonucleotides. The PCR revolution happened in 85.
Wow.
Which was, a year after I had left my PhD program.
It's crazy to think about the number of different molecular biology innovations required for a company like Nucleus to exist. I mean, it's insane to think about. Anyways, keep going.
Yeah, and there was the classic 85 paper in Science from the CETUS group, on PCR. There was the methodological paper with Mollison Faluna. And then in about 86, we in the lab started doing it. I was in an immunogenetics lab. I was interested in the immunology of the immune system and the genetics behind that immunology. And I remember the very, very first PCR. We had no thermostable polymerase. So every time you put the polymerase into a reaction, it died as soon as you
want to explain to people what a polymerase is?
Well, a polymerase is an enzyme that copies DNA. And the magic of PCR was you had a whole bunch of primers that were specific to...
Primers, want to explain?
Primers are short lengths of synthesized DNA sequence. And typically at that time, we were using primers of 17 bases to 20 bases. We would have one at each end of the gene to ensure specificity. Then we'd use a polymerase to make millions and millions and millions of copies of that particular target that you could then sequence, you could then hybridize, and you could clone if you wanted to clone it. Now, the very, very first PCRs were done with no thermostable polymerase. So every time you heated a reaction to 95 degrees to separate the DNA strands, you killed the enzyme. So every single cycle after the annealing of the oligonucleotides to the DNA, you had to put a new slug of enzyme in. And so PCRs took 4.5 hours. They were all done manually. And, we used to take a tea break in the middle and we'd have to hand it over to somebody else because you'd be on this cycle of, just continually putting polymerase in until I think it was Cetus invented the first thermostable polymerase, and then you didn't have to do that anymore.
So it's interesting because as you're describing your different PhD work, you can see already intuitively, the audience can see that genetics at some point was very much a wet lab science. And then over time became a computational science. And so I think you're kind of an encapsulation of this bridge, right? Which is really interesting, right? Because when people talk about, statistical genetics today, we're not talking about gels or, even the wet lab. We're really talking about computation. So it's just funny to hear about how, what a PhD even means in the context of genetics has just transformed.
Right. And Kian, it wasn't just wet lab. It was completely manual.
Yeah.
Because, as you know, so many of the wet lab parts are automated.
Automated, yeah.
The pipetting, it's done at scale. It's done at, in 96 well plates, 384 well plates and higher. The scale is extraordinary now. And remember, when we started sequencing DNA, there were no automated sequences.
Yeah.
And these automated sequences produce billions.
Well, we start with, Sanger sequencing, right?
Yeah, well, there was
automated manual.
There was Maxim and Gilbert sequencing, and Wally Gilbert was received a Nobel Prize for Maxim Gilbert sequencing.
Yeah.
And there was
Sanger,
and there was Sanger sequencing, and Fred Sanger received the Nobel Prize.
Yeah, he didn't know
for Sanger sequencing. And sorry, Wally, and he's a good friend of mine, but Sanger sequencing is so much easier than Maxim Gilbert, and I've never done Maxim Gilbert sequencing.
That's funny. So then, okay, you're in this kind of immunology genetics lab. You're running gels. You're traveling around the world, it seems, especially in India, to understand human genetic diversity. How do you get from there to Myriad eventually? Like, explain a little more. Like, how?
Yeah, that
how
was
to
a step. There was a
what happened?
What happened was a bunch of people I knew started Britain's first genomics company.
Okay.
And it was called Gemini Genomics. Okay. And it had a very simple idea.
What year is this?
This year was 1998.
Okay. So 1998. So was it Myriad started in the 80s?
Myriad was started, I think, in 92.
92. Okay.
Yeah.
Got
so
it.
this, yeah.
By Mark Skolnick.
Yeah, by Mark Skolnick.
Yeah.
Mark Skolnick.
So this is the first British genetics company. So Mark is in the United States. He's building Myriad.
Salt Lake City.
Yeah, he's in Salt Lake City. He's focused on BRCA testing, which we can talk about.
Well, Mark initially is focused on the discovery of the breast cancer.
Yeah. So he actually discovers BRCA1, BRCA2, one of them, and then he actually productizes it.
Yeah.
Correct.
Correct.
And so just so people have the context here. So basically, you have genetics in its infancy. You have I think, the beginning of the scientific genetic establishment starting to say, wait a second, there are sort of these, high penetrant genetic markers. There are these genetic markers that can cause something like, breast cancer, for example. And so people, intuitively, they know back in Gregor Mendel and his piece, there was this idea that, a single gene could drive a phenotype, right? And so a lot of genetics theory was built around this idea, what's called like monogenic analysis, or this idea that there's genetic markers in genes and they cause, a disease, some sort of debilitating disease usually. And what ended up happening eventually, someone like Mark Skolnick, who is a scientist, also the founder of Myriad Genetics, which today I believe is actually the largest clinical genetics company in the world. Mark and his team used different wet lab genetics techniques to identify and basically isolate the breast cancer BRCA gene, which is commonly used today to screen people for breast cancer, right? So,
Absolutely.
So I think he identified the BRCA gene in the 80s. Then he must have.
No, Mary Claire King originated the linkage,
Yes.
and so these various groups, Mary Claire King, Mark Skolnick,
Yeah, it was a bunch of independent groups.
They were collecting multi-case families. The multi-case families had multiple cases of breast and or ovarian cancer segregating in them. And then they went through with these linkage markers that existed at the time.
And so people understand this, right? Like, if imagine you're a scientist and you ask the question, like, what do I need to do to identify the genetic basis for, let's say, insert arbitrary disease, in this case, breast cancer? Well, a good place to start is to go to families where generation upon generation you find, wait a second, all these women are getting breast cancer. What's
Absolutely.
going on?
It looked like something genetic was happening in those families.
Exactly. And a lot of genetics, just so people know too, began familially. Right?
Absolutely.
Like these population studies, there's kind of a new kind of paradigm, but it began, hey, this thing runs in families. What's going on?
Yeah. The first positional cloning, which was what this technique was used, was done in families. And it needed sets of markers. And those markers, initially were RFLP-based VNTRs, variable number of tandem repeats. And I haven't said that acronym for a long, long time. They moved to PCR-able dinucleotide repeats, trinucleotide, tetranucleotide repeats. I think it was Lander and Skolnick first had the idea of these polymorphic markers across the genome actually being used for linkage mapping, and Mark was doing that. Other people were doing that. As I said, the linkage to BRCA1 was first recorded and published by Mary Claire King, and then Mark and his team competed with other teams to actually do the positional cloning, which is to bring the resolution down using these multi-case markers by recombination in families, and then physically identifying the DNA, using various libraries, back libraries, pack libraries.
And so you have Mark identifying BRCA, starting Myriad Genetics to actually start selling.
No. It was as he tells me, he started Myriad Genetics to win the race.
Oh, okay.
So
that's actually interesting. I didn't know that.
Yeah, he they raised capital
to actually win the race and
the
then productize it
to win the race, and so they had no guarantee of winning the race.
So by the time you joined Gemini, what was the name of the company?
I advised Gemini. That was 98.
So by the time you were advising, you joined Gemini, or
no? I had an academic position, and I think one of the really positive things about the British academic system is they let you go off one day a month and work with industry.
Okay, so you were kind of an academic, you were consulting with Gemini Genetics. At this point, I imagine Myriad Genetics is relatively more progressed. I mean, it's been only 6 years. They probably have started selling BRCA testing in some capacity, right? Is that true?
BRCA1 was cloned, I think, in 95.
Okay.
BRCA2 cloned and characterized in 96. Okay. And then the first product was launched in 96.
Okay.
Which was called BRCA analysis.
Okay. And so in the context of Gemini Genetics, they said, hey, let's also screen for breast cancer? Was that the...
No,
Yeah.
The model was slightly different. I'm just spend a minute on this because it's history. Gemini Genomics was founded to map genetic elements for a whole variety of phenotypes. So the idea there was you get thousands of MZ twins, thousands of DZ twins. You use the MZs and the DZs to figure out the heritability of a given phenotype, and it could be respiratory function.
Do you want to explain heritability to people?
Heritability is the proportion of added genetics variants that's attributable to genes. And so, we're all combinations. Every phenotype we have is a combination of, almost every, a combination of genetics interacting with environment. And so, there was this notion that the easiest things to characterize genes for are going to be A, the most monogenic, and or B, the most heritable. Seems reasonable. That's going to have the most signal.
So basically you're saying, hey, let's get a bunch of twins together and let's start understanding the genetic basis for various different phenotypes. That's basically what you're describing.
Yeah.
And was there like a specific phenotype that you guys were very interested in?
Everything you could measure.
I mean, that's kind of, that's a lot.
Yeah,
right?
Everything you'd measure. And so
what was the product? What were you trying to, was it like Skolnick-esque in that you were going to like actually conduct some sort of investigation and then productize something? Like
I think nobody at that point understood what the product was, but it was an incredibly exciting journey. It really was.
What happened? Did Myriad Genetics buy you guys?
No. They were bought by Sequenom. And so it was this, that I was going to say, it's the stuff of your dreams. It's not quite the stuff of your dreams. They were bought by Sequenom only a few weeks after doing their IPO. So they did an IPO in New York. Oh, yeah. Wow. For about $250 million.
Really?
Yeah. Which was a lot of money in those days.
Yeah, it's a lot, yeah.
Yeah, we're talking about 2000.
Wow.
And there were many unique things about 2000, as you might remember.
Yeah, I mean,
people were excited about everything.
Yeah.
They were excited about computers. They were excited about operating systems. They were excited about, online places that you might go and order a coffee or, buy a T-shirt or whatever. And they were excited about genetics. They were also excited about proteomics, but proteomics took a lot longer to be real. And so, they were bought by Sequenom, and Sequenom didn't really know what to do with them. And so the whole thing sort of withered away and died. For me, what it did was expose me to the US startup system.
Nice.
Somewhat to US venture capital, and yes, just the incredible opportunities that existed in the US. And, we all come here for different reasons. Mine was, goodness, that's where the gold is.
I feel like a lot of immigrants can relate to that.
Yeah. And so they should.
So, and so then you came to America?
Into 2002.
Wow.
Yeah.
Okay. And so you came to America and now you're like, hey, I don't want to be an academic anymore. I actually want to go into industry. Is that the model?
Yeah. And I would characterize that as I had 19 years of incredible privilege being an academic.
Yeah.
And interacting with just wonderful, bright people. But the big problem with academia is you can have the best idea in the world, and it's very hard to get money to explore that idea.
Yeah.
And I think it's a truism that because money is and always has been short in the UK, it tends to get concentrated in big academic centers. And if you're not in one of those big academic centers, it's quite hard to get the funding.
Yeah.
And so, I loved being an academic, but you tend to get narrower and narrower and narrower intellectually because you're only allowed to work on the thing that you're known for.
Yeah.
So I had a strong sense of that. I also had a strong sense that in America, if you had a great idea, somebody would give you money to explore that idea. And if it's a bad idea, you had to drop it really quickly. But there was just the instant nature of the ability to explore a good idea in industry.
It sounds like to me you're an entrepreneur. You said, okay, I did this investigation, I did this research, and now I'm ready to actually bring, and I think this has been true with you today, 20 plus years later, which is you have such a knack for taking technology, taking the most advanced research and saying, how can we actually make it useful?
I think that's true because, at Myriad, I ran a basic research group. But a basic research group is only as good as the product opportunities that it actually gives up.
Did you join Myriad then in 2000?
In 2002.
You came to America and you joined. So you came to America and then did you know you were going to join Myriad?
Yeah, they recruited me.
They recruited you. Yeah. So did Mark personally recruit you?
Yeah, he did. And in fact, there's a story behind that. I'll just tell that very quickly.
I just want to say this again to the audience who have context. Myriad Genetics is today the largest clinical genetics company in the United States, right? They've had a long history of selling clinical genetic testing. And so the founders, again, Mark Skolnick, who identified actually BRCA1, And now you have Jerry in 2002 actually joining Myriad Genetics, from his academic position. And so, yeah.
Why did I join? I joined because Mark wanted to retire. And he had already had one attempt with one person, which didn't work out. How did we meet? Interesting story. I was running a PhD project with a PhD student who's now a professor at University of Colorado. And that project involved examining immune parameters in people that we could genotype. And I once sat at breakfast with Leonid Krygliak, who is an absolutely brilliant geneticist. I think he spends most of his time on yeast now, but brilliant guy. I remember sitting at breakfast with him, and I said, Leonid, I've got this problem. I've got no money, but I've got some ideas of what to phenotype. What resource is there where the genotyping's already been done? And he said, well, there's the UGRP, which is the Utah Genetic Research Project. So these were multi-case families, which were being studied at the University of Utah for everything. And they basically sort of took the Gemini idea and phenotyped these people for everything, and perfect pitch, blood pressure, cardiac output, FEV, everything, every parameter you could measure in blood. And it was the same kind of idea, but in families where these phenotypes were segregating through these massive Mormon families. Because remember, the thing about Utah, it was originally peopled by Mormons. Originally, those Mormons had 5 wives, 10 wives, 15 wives, 25 wives, and they're very attentive to their genealogies.
Yeah,
which is why Myriad got started there in the first place.
Ancestry too, right?
Yeah, Ancestry is down there in Provo. Yeah, Ancestry is a very smart bunch of people, which is another story. You had these massive groups. So I called up the director of that project and I said, we'd really love to measure some immune phenotypes in your group. Have you been doing that? And they said no. So I said, well, let's come over and do it. And this gets a little bit entrepreneurial because we had no money to do it. So a bunch of people, myself, my wife now, and Paul Norman, who's the professor in Colorado, we would take weekend trips because the families came in on a Saturday to avoid the working week. We would get on a plane on Friday and we would fly over and we would take blood and would measure all these immune parameters on these people. And thank you, Carl Witwer, who was one of the pathology professors at university. He gave me a little corner of the lab to work in, gave me access to their machines. We had no money, and we bought those flights on our credit cards.
Wow.
And I look back on it, and it was so crazy. But we did it, and we ended up mapping a bunch of immune phenotypes.
And that's how you met Mark.
In these how did I meet Mark? I came over to pitch phenotyping in families to Mark, some other phenotypes as a project. And so I remember we were going to go to Bambara for dinner. And then I was ready for dinner. And then he phoned me up. He said, I can't come to dinner tonight. I'll go tomorrow. And he picked me up and we started talking in the car. And you know how it is. So we talked. We had dinner. We talked. And then it got quite late on Saturday night. And Sunday morning, I wake up and there's a call. And he said to me, can you get me your CV in half an hour? And I said, well, I wasn't looking for a job and I don't have a CV, but I'll bang something together. So I had my computer with me. I banged something together. I sent it to him and he said, well, we want you to come and work for us. And I said, well, I don't know about that. It's a long way from home. Nice place. I feel like I'm on holiday every time I come here. Mountains are beautiful. Yeah, I like to ski. I've been skiing a little bit. And so, we thought about that. And, he's an amazing individual. The group was extraordinary. The company was extraordinary. And eventually they made me an offer I couldn't refuse. So, I joined at the end of the year.
Okay. So
and that was something because I left behind tenure, permanent job for life, and all my research relationships. And, some people were quite upset that I left academia to go to a commercial company. But that was the next phase of our lives.
Do you know what was Myriad's market cap at that point?
Quite small, actually. I know they had 2 million in revenue.
Okay.
And that maxed at about 800 million.
800
eventually.
million.
Yeah. It was
So you probably took the company from, let's say, I don't know, $20 million to $2 billion?
Yeah. They were 4 at one point.
4 billion?
Yeah.
Okay.
Yeah.
Let's talk about
But can I just go back to, but the idea was come in, do the best basic science you can, but always with eyes on a product.
And I think they give you an offer you can't refuse, because I cannot imagine a better candidate for that job than you, because you are always thinking basic science, getting the fundamentals right, having that scientific rigor, and then thinking, how do we productize it?
Yeah.
Right?
Yeah, and I think that's right. It's very gracious of you to say that. I think that's right, and I was lucky enough to work with a team who shared that priority, and were.
And by the way, for how long did you work at Myriad for?
19 years. Nearly 20.
Wow. So you did 19 years in academic, then 19 years as a chief scientist?
I was senior vice president of research, and then executive vice president of research, and chief scientist in 2010.
Okay, so I want to then talk about, I mean, you've seen genetics, basically.
Yeah.
Your career is an encapsulation of genetics, and one thing that I think always comes up in our scientific advisory board meetings is there's genetics, there's science, and then the difficulties that you can run into trying to bring very good science, very rigorous science to the public. And the thing I want to highlight here is I want you to tell the story that you've told me, that Mark has told me, about the original BRCA testing.
Yeah.
Right. So today, BRCA testing is very well established. Yeah. Right. Hey, a woman wants to know their genetic risk of breast cancer. Here's a BRCA test. I mean, people don't really bat an eye. I think it's very much commonplace.
Yeah.
Let's go way back in the day, though. You said, Myriad started in whatever, 1996 or they're like
92.
92. Okay.
Product was launched in 96.
Products launched in 96. Right. You're bringing BRCA testing, you're bringing genetic testing to the clinic.
Yeah.
Right. And the reason why I wanted to go through this, because I think there's many parallels between then and now, and the story going forward in genetics. So can you tell us that story? What happened when you tried to bring BRCA testing to the clinic?
Yeah. A couple of things. There's resistance from much of academia, because much of academia felt that women were not yet ready for this information.
Wow.
And there's always this worry of psychological harm, that maybe people don't understand the consequences. There are familial consequences of being a carrier, as we know. And so there was some sense of unease in the academic community. But that didn't matter because the cat was out of the bag. And so women in these families had seen their grandmothers, their aunts, their sisters contract breast cancer, die of breast cancer, die of ovarian cancer, were desperate to get hold of this technology, because it was life-changing. And why is it life-changing? Well, in the Mendelian context, where you have a very, very high penetrance mutation, you have 3 daughters. One of them could have inherited it. The other 2 might not have inherited it. But as you're able to test, you're able to tell those 2 daughters that haven't inherited it, you're in the clear. You don't have to live with this sword of Damocles of breast cancer over your head. With the woman that has inherited it, there are preventative measures that she can take if she's carrying one. There's mastectomy, which is, it's a serious surgery, but it can prevent a woman getting breast cancer. There's chemotherapeutics like tamoxifen they can take as a preventative, which halves the risk of breast cancer. It doesn't eliminate it completely. Or they can have regular screening. They can have MRI, mammogram, 6 monthly, and they'll detect it early before it's disseminated, before it's metastasized. And so all of those things are options which different women and their physicians choose. But the point is, you create information and you can create choice that is based on that information.
So we see a genetic test, in this case BRCA testing, based off very rigorous good science, can help transform an entire life, especially for a woman.
Yep.
You see resistance from academia.
Yep.
How about, doctors? How about the NH kind of, the NCRI? I'm not sure if it existed at that point, but what were the Human Genome Project people saying, right? What, like, when you did this, what was sort of the, academics, they may be saying I'm skeptical, but how about the clinical community?
There are always early adopters, and they were all over it. They wanted this tool.
The patients
wanted the tool.
How did the doctors feel?
Many doctors wanted the tool.
Okay.
Many doctors and this is an emerging and continuing theme, many doctors weren't up to speed with the new genetics.
Okay.
And we can talk a little bit about that later as well. Many were. But what are the bottlenecks as you try to scale this kind of life-saving technology?
Yeah.
And remember, it's not just breast cancer, it's cardiovascular.
Yeah.
And
were there any stories earlier on that you think encapsulated the potential resistance of bringing and empowering patients with their own genetic insights?
Well, one of the biggest impediments is getting this paid for. And so, it's a little like the Uber story in many ways. Ubers were subsidized around the world to get people into cars to create scale. If your genetic test was not being covered by insurance, you had to pay.
How much was it?
I think it was about $3,800 when it started.
I mean, how crazy that is. $4,000 approximately for a BRCA test.
Yes.
And we have a whole genome test today for $500.
Yeah, I know.
I mean, what?
I know. But
people understand that, like, BRCA's one gene, right? BRCA1's one gene, BRCA2's one gene, right? Wait, is it panel already?
Well, let's
is it like a specific? Is it like 3 markers? Like, what is it?
Well, it's developed. But let's go back. If you go back to 1996, the whole genome had not been sequenced.
Yes.
The cost of doing a genome at that point was $3 billion. Yes. And you've talked about, when it was $10 million. And one can look at that curve, it's like the Moore's law curve going down. So, back in 96, it was expensive to sequence DNA. And you had an insurance system, private insurance system, certainly Medicare, that was not ready for that testing. And so it was a real slog to get insurance, authorization and payment for that. And you talked about what was being done at the beginning was, BRCA 1 and 2, and then large rearrangements were added, and the database behind variant classification became richer and richer and richer with numbers and with scale, which had tremendous value. In about 2011, 2012, those panels were expanded, and there are now about 13 high-risk, intermediate risk, inverted commas, Mendelian genes that are screened for breast cancer. It's around that. The numbers creeping up. So panels started to be done, and after the Supreme Court case that Myriad lost, those panels were offered by many, many different types of tester.
Want to talk about that quickly? That's actually interesting point.
Yeah, if you like.
Yeah, go ahead. So basically, at some point, Myriad famously tried to patent.
Did patent?
Well, you did patent. Tried to keep the patent, and that's when you guys got sued, or what happened exactly?
Yeah, what happened was the ACLU got very interested in this case, and there's a book about it. There's probably more than one book about it. There is one exceptional book about it. And let's put that in the show notes.
Okay, sure.
And it went to the district court, Southern Court of New York. Is that what you call it?
Sure.
Well, anyway, they had this wonderful court in New York, and I think Judge Sweet heard the case and decided that these patents shouldn't exist, shouldn't be.
How do you feel about that?
Continued. How do I feel about it? I feel that he made the right decision for the wrong reasons.
Okay.
And it didn't matter much at that point, because I think at that point the patents were about 18 years old and they run out at 20 years. So it didn't make much material difference. That the Myriad went to appeal, and the appeal court is 3 federal judges, and they, I don't know if quashed is the right term, but they quashed the original finding of the Southern District of New York. And then famously, the case went to the Supreme Court and was argued in the Supreme Court. And the Supreme Court came down against the patents based on their judgments and their wisdom, and based on some interesting analogies, which are all played out in the public record.
Okay, well,
yeah. So that's the short story, and then anyone could test. Subject again to CLIA and CAP and the clinical regulations, and then I think we started to see just an explosion of the technology. I think it had exploded with Myriad in the first place. Millions and millions and millions of women had already been screened, and so that was one product, as you know.
One thing I want to talk about here, which I find very interesting, is we're talking about BRCA testing because Myriad is very famous for that. That's how the company started, and eventually they expanded to all sorts of different panels and all sorts of different kinds of hereditary genetic testing, right? And now they even have polygenic testing and beyond, right? What I find remarkable is I go to a fertility clinic today.
Yep.
Okay. The couple, they'll look at me and they'll say, "Yep, we ordered our preconception testing," and I'll say, "Oh, do you have a family history of breast cancer?" They'll say, "Yes." Right, breast cancer is a pretty common condition. As we know, 15% of women approximately have breast cancer, get breast cancer in lifetime. And, they'll say, "Do you have a family history of breast cancer?" They say, "Yes." And then I say, "Well, that preconception panel you ran, it doesn't include BRCA."
Right.
What gives? I mean, it's been 20 plus years. We know that BRCA markers are high penetrant. They still maintain high penetrance, right? We know that they have a profound consequence, not just on the woman's health, but also on their future child. And it's cheap enough now, right? You can run a whole genome $500. What is going on, do you think? It's kind of weird to me that, in a way, BRCA testing is archaic, but I go to the IVF clinic today and it's still somehow a frontier test. Help this dissonance.
There are a number of different problems. One is within the clinical sphere, a lot of the testing is siloed. It fits into particular clinical environments, particular clinical disciplines, and it's insured and paid for in particular ways. And all of that is very narrow and often not very effective, and I could give you some examples of that. But it's siloed. Now, if I can go back to another thing, you mentioned familial breast cancer. So the thing that was really obvious to me in 2014, 2015, was that the field was doing a partial job in characterizing the genetics of breast cancer. And there have been just some amazing public consortia who've run enormous GWASs on genomic whole association studies, and hundreds and hundreds of thousands of people. But that information was not being translated into the test environment, into the care environment, and wasn't being offered in a coherent.
So you're talking about polygenic scores.
Yeah, I'm talking about, the other half.
And so
breast cancer.
You were saying BRCA testing in the context of a preconception environment wasn't screened for, and then you're now talking about another kind of way of analyzing DNA, which is polygenic scores, because it turns out many of these phenotypes, like breast cancer, actually weren't just driven by BRCA.
Absolutely.
There were actually many, even up to 1 million genetic markers that shape a woman or a man's risk for breast cancer. And then you can apply that logic actually to any phenotype, right? Not just breast cancer,
Absolutely.
but, height and coronary disease and schizophrenia and Parkinson's and MS and beyond. And so you're saying that then there was, in a way, then we're touching on something interesting here, Jerry, which is there's research,
right? High
quality, rigorous research, but then there's this lag time to bring it to the marketplace.
Yep.
And, I hear the insurance argument, but what else is going on here? It is astonishing to me as someone who, came to the genetics industry from my bedroom, so to speak,
Yep.
right? That's how I started. I started wide-eyed and saying, I want to bring the best technology and the best information and put it into everyone's hands.
Yep.
And what I find most surprising is the resistance to do that. It's never the patient, by the way. It's never the patient. It's researchers. It's sometimes even physicians,
Yep.
right? And it's not a cost issue anymore. We know that, right? And I just, you've been in this space for 20 years. I want to understand from you, when you hear that a BRCA test isn't part of a preconception testing panel, to me, my interpretation is someone somewhere is making a decision, a very arbitrary decision, that, hey, we shouldn't, in the family planning context, even factor in what's called autosomal dominant conditions or later onset conditions, right? So, and so just so people understand this, usually when you do family planning, classically, you would screen for things like cystic fibrosis, which is like a rare condition that's caused by a single gene that's really, really bad. Then there are conditions that are a little bit later onset, like breast cancer, for example. You don't get breast cancer when you're 5 years old, but you get breast cancer later on. And so, the idea of screening for these conditions, many of the conditions, by the way, which actually kill people, coronary artery disease, cancers, right? We talked about, schizophrenia, common conditions. These conditions, which are screened later for in life, generally the kind of opinion in the preconception planning context is to say, hey, we don't need to screen for those, even though really the marginal cost is negligible, right? And when you go and you actually tell patients, hey, you can screen for hereditary markers that are autosomal dominant that have profound impact not just on your health but your future children, they are so shocked to find out, okay, that gene or genes are not in their preconception panel. Help us understand this. Why and what can we do to solve this?
Well, I think there's several aspects to this. One of the aspects is what you are allowed to know, which is very interesting. And you go back to ACMG, for example, the American College of Medical Genetics. They have this list of things you're allowed to know in secondary findings, for example. So you can go in and have a whole genome done. The community is really worried about you knowing this thing that you didn't know about when you went in for your test. Secondary finding. You've gone in to have a BRCA test, and they've discovered you were an ApoE4 carrier, for example. You didn't ask to be an ApoE4 carrier. We now know you are, perhaps at the clinic level. Do you need or do you deserve to know that information? And that's not picking on ACMG, but there's this sort of control of knowledge aspect, which is, to my mind, incredibly paternalistic. And just to digress for one moment, our DNA is our story. We have 3 billion base pairs, and I find it extraordinary that there are people out there who believe they should determine which of those bases you know about and which of those bases you don't know about, which of that information is useful for you to know, and which of it is somehow too dangerous for you to know. So that's the first thing. It's your story, and to my mind, there is a part of the world that's shielding 200,000 base pairs of your DNA to take an arbitrary number, and you're allowed to know about the rest of it because they don't particularly care about the rest of it and they don't think it's dangerous. So that's one thing. Secondly is how things are paid. And classically, clinical genetics is quite expensive because you're paying for a bunch of experts. You're not just paying for a DNA sequence. You're paying for all sorts of expensive people in labs, that are regulated, who have to sign off on things. You're paying for commercial organizations that have to go out and sell that product. You're paying for marketing. We all know how expensive that is. So you're paying for all of these things. So you've got the control of information, you've got the payment of that thing, and until recently you had the technology. We've been able to do panels fairly cost effectively for 10 years, say 13 years. Now we can have genomes done extremely cost effectively. Now, I know that there's this argument that, all you need is a short read pass of the genome and you've got everything. And I think you'll know that I don't agree with that. And, when we talk about the technology stack, and actually the genetics stack, there are quite a lot of layers. Long reads are incredibly useful for haplotyping. You can get to understand the HLA system by using long reads. You can't really construct it probabilistically from a random bunch of short reads. Large rearrangements are a relatively unexplored area. Each of us has got thousands and thousands of big and small genetic deletions around the genome. And because they haven't been looked at terribly carefully, we don't really understand the clinical consequences of those. And so you've got dosage issues. And, again, we talk about scratching the surface. We are starting to scratch the surface there with those kind of entities.
The analogy I like to do sometimes is like, I like to give people an intuition of this is like we are exploring the genome in the way that we're exploring like the moon, right? Like we've been there, we kind of mapped it out a little bit, but fundamentally no one's there, right? Same with the genome. It's like it's the very beginning, just like how we're exploring our own solar system. We're exploring our molecular system as well.
Yeah.
And people should treat our understanding of DNA as a new frontier. It's an intellectual frontier that's going to dramatically improve. I mean, think about in the last couple of decades, right? We're talking about manual lab processes. We're talking about a $3,000 BRCA test, right? To today, you're running a whole genome test, for $500 at scale. And in other words, you're talking about going from like a unicycle to a car, in what, 15, 20 years? I mean, it's extraordinary. It's an extraordinary technological achievement. And I think part of what I'm hearing from you is there's been this extraordinary technological development, and now there's this question of like who gets access to what information, and that's where we kind of go from science to sort of politics, right? I think that one of the things that we always stand by as a company is that the patient fundamentally has the right to their own information, and I know this is something that's very near and dear to you, right? You really believe, both in the adult context and the embryo context, right? Patients should have all there is to know about their genome. Want to talk a little bit more about that?
Yeah, I do, because and I think you can think about this historically. For example, all of those patients with family histories of breast cancer, family histories of ovarian cancer, family histories of pancreatic cancer, they very naturally went into the clinical, and this was it was a symbiosis. They went into the clinical testing environment, and the clinical testing environment blossomed because of those families coming to them. And the services those families were given were, I think, incredibly powerful, potent, and helpful for their healthcare. A lot of women, a lot of men, in fact, the vast majority walked away from that without a result, without a genetic result, until polygenic risk scores were introduced into the system. And at that point,
want to explain just polygenic scores again for people?
Yeah. So if we go back to 2015, there were really two worlds. There was the world of high risk, high penetrance, intermediate penetrance, Mendelian mutations that underpinned ovarian cancer.
So really bad
cancer,
genetic markers, any gene.
Really bad family histories. So they existed. The academic community was involved in looking at millions and millions of GWAS SNPs. So they were doing this with microarrays.
They found genetic markers that are disproportionately associated in a group of people that share a common disease, for example.
Yes, and that's called a population association. When you run those through cases and you run them through controls, it's the science of population association. But those markers weren't really going anywhere. Academics love them, do fantastic work. They're never really sure when that work's over, because numbers can get bigger and bigger and bigger, and you get more and more sensitivity, and you get more and more power, etc., etc. Nothing's ever finished. But there comes a point where you've got to do something practical with that information. So my group and I set out to do something practical.
Myriad.
Yeah, at Myriad. Set out to do something practical, and that was in women who aren't carrying those Mendelian high-risk, high-penetrant mutations. Were we able to offer information that informed their breast cancer risk? And so we put together really the first clinical polygenic risk score panels for women.
Let's break this down. So let's say there's 100 women. How many have a BRCA variant? 3? 5?
Well, it depends if they've got family history or not.
Okay, let's say just general population.
100 women,
general
about
population.
0.5% will have either a BRCA1 or a BRCA2 mutation.
How about any intermediate variant like CHEK2 as well, or PALP2?
CHEK2 is commoner, and it's a low prevalence. So about 1% will have CHEK2.
Okay, so let's just say across all the genes.
PALB2 which is at the other end.
Yeah.
About 0.1% of those women will have a PALB2 mutation.
So let's say you take a panel of around 15 strongly associated or moderately associated breast cancer related, ovarian cancer related genes, whatever. Let's just say a couple people out of 100, right? 2 to 5, would you say?
Yeah, depending on the family history. The stronger the family history, the higher that frequency will be.
Okay, so let's just say in a general population where you're not necessarily selecting for family history, it's a couple people. That's fair, right?
Yeah, I think 1% ATM, 1% CHEK2, and
sure.
0.5% BRCA.
Great. So let's just have 100. Let's just say 3 people. That means 97 women who used to do old school genetic testing, if you will, old school genetic testing. The doctor would say to them, well, we didn't find this, BRCA marker, we didn't find this CHEK2 marker. I know this, we didn't find a breast cancer marker. And so your risk is probably 15% because it's the lifetime risk. We don't know. In other words, they don't have further information to stratify the risk of the remaining 97 people. But then what scientists realize is actually there's up to 1 million other markers now that do shape a woman's risk of breast cancer. So it turns out that BRCA testing was the first step, so to speak, and that to now stratify the risk, and by the way, the risk of the people with the markers, but putting that to the side for a second, but to stratify the risk of the remaining 97 women, you should actually run something called polygenic testing.
Yeah.
And polygenic testing, we know in breast cancer, it's very strong, it's very robust. I mean, you want to talk about that? I mean, your group was the first group to bring a clinical grade polygenic score combined with BRCA testing in a clinical environment. I want to talk about that?
Yeah, it was. And it, beyond BRCA testing, so it would have been a full
panel.
Hereditary cancer.
Probably what? How many genes? 10, 15?
Around about 26 at that point. So yeah, it's the other half of the equation. There are women without the high-risk familial genetic markers who still have a high risk of familial cancer. And many of them are up at the high end of the scale on a polygenic risk score. And a polygenic risk score just measures many, many, many small elements throughout the genome and then combines those mathematically and places you on a scale of risk. Now, for breast cancer in a woman of European background, they have about a 12.5% lifetime risk of developing breast cancer. And so you can play, and we'll deal with genetics first and then slightly elaborate on the model. You can add a whole bunch of other risk factors together, of which family history is one, but you can put in BMI, you can put in ethnicity, you can, height's obviously a component of BMI, whether they drink alcohol or not, whether they've had hormones or not, whether they've ever had a breast biopsy or not, a whole bunch of clinical and biological information, and produce, a really pretty good predictive model in women who aren't carrying those hereditary mutations. And that kind of model, in my opinion, should be available to all women, not only those in families. And so, the testing companies, because of the way that all of this is paid for, tend to offer those kind of products to women with insurance and have it paid for insurance. And somebody, maybe can let us know later. To my mind, the polygenic risk score models are not yet paid for by insurance. And Myriad launched theirs in 2017. It was updated in 2021 with a model that works for women of all ancestries, and kudos to the team. I'm really proud of them for doing that. It was a long haul. It's an incredibly difficult scientific project, but they did that. So out there you have these models that are available for people of any ancestry and mixed ancestry. And they're not in people's hands because they still sit within the clinical testing environment, and they're almost rationed to people with family history. And then just, one little vignette, and this is probably obvious from what we've talked about. Women with family histories of these conditions are enriched for these genes, but they don't just exist in women with family history. And so there are as many BRCA1 and 2 mutations in women with no family history of anything as there are in women with a family history of breast or ovarian cancer. Wow. And so half the population goes completely unscreened for these potentially lethal, life-changing genetic elements like BRCA1 and 2. And one of the reasons I'm so excited about Nucleus and other consumer testing entities is you've got the ability to go out and capture that other half of the mutation pool. And in the US, that's, 1 million people who are completely unaware that they're carrying this. And I think one of the exciting things about where we are now and what we're talking about is we're not just talking about these 1 or 2 genes with these mutations. We're talking about getting an entire genetic life screen for a few $100s. And I find that transforming.
Yeah.
I find it empowering.
Wow.
And it's just a great topic of conversation and a great practical thing to do.
I completely agree. I think it's also just going to get cheaper and cheaper and cheaper and cheaper.
Right.
And that's another thing that I think is just going to be a amazing accelerant of the technology. I think we'd be remiss to not talk about embryonic selection,
Right.
at least briefly.
Right.
We recently launched a Nucleus Embryo. And, I think we got over 12 million impressions. It resonated. It resonated, I think, with couples all over the world. I mean, today I did an interview with, I think, one of the largest publications in Turkey. We also had outbound from France as well. And so I think it's just like, there's a lot of international patients that reach out to us as well from all over so many different countries. And so, I'm curious as a company like Nucleus comes and, we are sort of thinking about actually the underlying technology that we use to analyze adult DNA. It's actually the same as embryo DNA, right? Really, practically, it's like the same, because where do the models come from? Well, they come from adults, right? And so I'm curious for you, and seeing the way genetics has evolved, now seeing this kind of genetic stack forming from adult testing through embryo testing, embryo selection. Does it excite you? What did you think about Nucleus Embryo? Tell us your thoughts here.
Yeah, it excites me very much, and it excites me from a freedom of choice perspective for parents. And yeah, I heard a term the other day that I really liked, which was grumble groups. And I don't know if that's a general term or if it's very specific to a generation. But there are all these grumble groups who worry about particular things, and I think my touchstone to deal with this is freedom of choice. And freedom of choice bears now upon a whole range of genetic elements and a whole range of phenotypes. And so, there is a group of people that don't believe that there should be any interference in this process, that, the passing on of an hereditary cancer mutation should be left in the hands of God, for example. And I don't believe that. I believe parents should be armed with the best possible information to ensure the health of their children and the ability to choose health for their children. So I think that's controversial to one grumble group, but not generally controversial. So I believe in an absolute freedom of choice. And if we think about abortion, for example, there are some states in the US where there isn't that freedom of choice for abortion, the woman's right to choose. And there are some states where there is much more choice on that topic. And sort of if I take that as an analogy, embryo selection is something that happens upstream of a difficult decision like that. And it seems to me it's a modality that enables you to avoid difficult decisions. We've talked about choosing an embryo, for example, that doesn't have a cancer gene. We can choose an embryo, obviously, that doesn't have a trisomy 21 or a trisomy 18 or trisomy 13.
Yeah.
So again, that would obviate the difficult choice of abortion downstream once you did NIPS or NIPT or you did an amniocentesis or what. All of that sounds to me pretty uncontroversial. So I think chromosomal anomalies, pretty uncontroversial. Chromosome imbalances, pretty uncontroversial. There are some interesting arguments to be had about deletions in the DNA and the size of deletion, things like DeGeorge syndrome, when that gets really small, what are the consequences of that? And classical DeGeorge is associated with, I think, an average 40-point drop in IQ. So, that's genetic deletion that bears upon IQ and a whole host of unfavorable phenotypes. Is that controversial? For some, maybe not for others. And then we go, all the way, to traits. And I know that you are able to offer some probability around traits based on genetic information. And, really, what's so bad about being able to choose blue eyes or, you've got beautiful brown eyes.
Oh, thank you.
Wow. I love brown eyes.
Oh my God. Wow.
Why, is it controversial that you should be allowed to choose a couple of children with brown eyes and a couple of
You
children
pick your
with
partner
blue eyes
already, so.
Yeah. So, so I don't find any of that controversial at all. And then you get to the sort of other end of the spectrum where, to me, many of the models are less explanatory because they're newer and they have less data. And it's a question of, is it controversial to offer information that's perhaps a little shakier or a little less wobbly, a little more wobbly, or a little less discriminatory? And again, I think it's a matter of choice. But I think in that situation, parents should understand how powerful the models are and what the limitations of the models are.
I agree 100%. Okay, I want us to end on something. I think, one thing I think a lot of people don't know about Nucleus is how dynamic the scientific advisory board meetings are. Oh my God. We have amazing breadth and depth of experts. And I joked with you before this podcast. I was like, imagine that was a podcast. I want to maybe ask you a question I would ask you on the advisory board. So I would ask you the following, which is, I want you to tell me something that you think we're doing well and we should continue to do, and something we're doing badly. Okay? You can start with everyone and, let me know. We can talk about it.
I think what you're doing extremely well is bringing a product for a very reasonable price to anybody that wants to sign up for it, to give a depth of knowledge of their genome that they can't readily get in any other context. And, I would applaud you for that. The second thing I would applaud you for, and I'm completely honest about this, your product velocity is staggering. And I've complimented you before, and I've complimented you in public on that. It is truly staggering, and kudos to the team. I'm blown away every time. I'm blown away to the fact that what happens, and people won't know this, but you tell me, Jerry, we're launching this product on Monday that I don't even know about. And there may be reasons not to tell me about it. But I'm completely, and I stand back and think, oh, that's pretty important and pretty interesting. And they've done that really rapidly. And so I'm sort of blown away by that. I love working with smart, intelligent, dynamic, energetic, fun, nice people.
Yeah.
And if you weren't nice people, we wouldn't all have the relationship we have, and I don't think we would have half the fun we have.
Yeah.
So I think you're absolutely great in all of that. I think any critique I would have is my dream and my goal is that this technology should be available to the poorest of the poor. And I say that driven by one reason, and that is there is no family that cannot afford the burden of a preventable disabled child any more than a poor family.
Yeah.
And poor families right now do not have access to this technology. They don't have awareness of this technology. They perhaps lack the education to appreciate it. And to me, the medically driven and medically relevant aspects of this technology should be available on Medicaid. It should be available to the poorest of the poor. And I look forward to a time when there's true democratization of the technology, because it costs virtually nothing, or it's paid for by some foundation, or it's paid for by government, to truly make this available to the people that need it most, and not just the Silicon Valley billionaires and millionaires.
Absolutely.
So it's a fundamental for me, and that's what I'm working towards.
Well, I think that's what we're working towards. I mean, I couldn't agree more with you. The ethos of a business like Nucleus is to put genetic testing technology into every single person's hand, no matter where they're from, no matter how much money they have, no matter what they look like. Every single person deserves access to genetic testing. And if you think about it, I always like to give the analogy of the smartphone, right? At some point, the smartphone was in, a small number of investment bankers' hands. They said that to me, it's correct. Yeah. And then today you can see, billions of people have a smartphone. And if we follow that model, and you follow the model of what a successful enterprise, what a successful startup looks like, it looks like inherent to the DNA of the company, if you will, is democratization, right?
Yep.
And so I think I completely agree with you. And I think in the next 5 to 10 years, you're going to see a dramatic exponential adoption of this technology, not only, by the way, through consumer channels, but through, I think, clinical and government eventually channels as well. And I think that, to your point, we as a company are so keenly aware, from the advisory board to every single one of our employees, that when we show up to work every day, the number one thing on our mind is, how can we help another patient? And I think both of us stand for that, and you've been doing it much longer than I have, and so I appreciate you. Thank you for all the way you've helped serve patients, and of course, thank you for all the ways you helped Nucleus, and thanks for coming on.
It's been a pleasure and still remains a pleasure.
Thank you, Jerry. Thanks.